Multiple sclerosis, MS, is an acquired immune-mediated inflammatory condition of the CNS resulting in areas of demyelination, gliosis, and secondary neuronal damage throughout the CNS. MS usually develops and showing symptoms in people 40 years. MS can be classified into 1. Relapsing remitting MS, RRMS. Most common form, episodes of symptoms, relapses, followed by partial recovery, remissions, then periods of stability, resulting in residual damage remaining in the CNS, leading to SPMS in 60%. Secondary progressive MS, SPMS, occurs as a consequence of severe PRMS attack that's silently and gradually accumulating disability through time, which become less frequent or stop completely. 3. Primary progressive MS, PPMS, less common type, characterized by gradual and steady progression of disability, without remission yielding constant and gradual deterioration, 15% of MS cases. 4. Progressive relapsing MS, and PRMS, the rarest form of MS, characterized by steady decline in cognitive and physical function with acute relapses that occur repeatedly over time during disease course. Multiple sclerosis pathophysiology. Experimental lab studies in human samples, CSF and blood, have revealed the essential roles of adaptive immunity through cytotoxic CD8 plus T cells and T helper CD4 plus cells in the development of MS. Leptomeningeal, PIA, and arachnoid meninges, inflammation, disturbance in the blood-brain barrier, and abnormal vascular permeability are important factors for BBB disruption and hence leukocytes transmigration into the CNS. The transmigration of pathological leukocytes into the CNS is mainly mediated by interaction of integrins such as lymphocyte integrin, A4S1, VLA4, expressed on the surfaces of leukocytes with their ligands, cell adhesion molecules, CAMs, such as VCAM1 and P-selectin, expressed on the endothelial cells. In response to CNS injury, microglial cells become activated and macrophages infiltrate, leading to 1. The activation of myeloperoxidase, MPO, enzymes that induce inflammation and degeneration. 2. Release of reactive oxygen species and nitric oxide that induce mitochondrial dysfunction, leading to oxidative stress, lack of ATP supply, activation of acid-sensing ion channel, ASIC, and consequently demyelination, apoptosis of oligodendrocytes, and degeneration of axons. 3. Demyelinated regions encounter reorganization of sodium channels and hence accumulation of intracellular sodium ions, thereby promoting the sodium-calcium exchanger to operate in reverse to induce calcium influx. 4. The rise in intracellular calcium activates further sodium channels that induces calcium release form cellular stores, which can damage mitochondria, activate nitric oxide synthase, and harmful proteases and lipases, leading to neuronal cell death. 5. Increases extracellular concentration of glutamate that can overstimulate glutamatergic receptors and hence causing neurotoxicity to neurons, oligodendrocytes, and astrocytes, and inducing gliosis. 6. Induction of matrix metalloproteinases, MMPs, involved in fragmentation of myelin basic protein, leading to the generation of immunogenic fragments and demyelination. 7. Iron is liberated from degenerated oligodendrocytes, activating microglia-induced oxidative injury. 8. As a consequence of neuronal damage and oligodendrocyte apoptosis, astrocytes hypertrophy and proliferation seen within and at the margins of inflammatory demyelinating lesions, and also in normal-appearing white matter, gliotic response, that might limit the repair mechanism of remyelination. Multiple sclerosis risk factors. Based on in vitro, in vivo, and clinical data, inflammatory responses have been documented as the main cause of MS. However, the exact cause of such hyperactive and uncontrolled immune activity has not been identified yet. The etiology of MS remains unclear, but the hyperinflammatory response has been linked to numerous risk factors. 1. Gender. 2. Genetics. 3. Environmental factors. 4. Infections. 5. Fibrinogen. 6. Toxins. 7. Trauma. 8. Obesity. 9. Smoking. 10. Microbiota. MS and gender. Women are more affected than men. Sex ratio 2.5.1. Preclinical studies revealed that tamoxifen administration results in the inhibition of oligodendrogenesis and new myelin formation while preserving existing myelinated circuits. Tamoxifen is estrogen receptors modulator, mainly acting as an antagonist, that showed inhibitory effect to new myelin formation. Therefore, estrogenic pathway may play potential mechanism in developing MS. MS and genetics. People with an affected first-degree relative have a 2 to 4% risk of MS, as compared with approximately 0.1% risk in the general population. The occurrence in monozygotic twins is 30 to 50%. Genome-wide association studies have identified more than 200 gene variants that raise the risk of the disease. Human leukocyte antigen, HLA, encoded surface molecules present antigenic peptides to T lymphocytes and play a key role in adaptive immune responses, physiologic or pathologic. Most risk alleles are associated with immune pathway genes, a finding consistent with the notion that autoimmune mechanisms for the disease, perhaps against myelin-related antigens in the restricting context of DRB1-1501, MS, and environmental factors. Major environmental risk factors include 1. Geographic latitude, with a higher incidence in more temperate climates, which may reflect seasonal changes in sunlight exposure influencing vitamin D levels. 2. Pathogens prevalent in these regions. 3. Genetic contribution in the populations of particular region. The active form of vitamin D induces monocyte proliferation and the expression of interleukin-1, IL-1, and cathelicidin, an antimicrobial peptide, by macrophages, contributing to innate immune responses to some bacteria. MS and infections. Clinical reports documented the genetic materials of numerous pathogens in MS lesions, including chlamydia pneumonia, human herpes virus 6, HHV6, and Epstein-Barr virus, EBV. Tonsil to germinal center, GC. Naive B cells are infected by Epstein-Barr virus, EBV, that proliferate in GCs. EBV-infected memory B cells exit the tonsil into the circulation. Normally, EBV-specific cytotoxic CD8 plus T cells control the number of EBV-infected B cells. In MS, EBV-infected B cells are not controlled by T cells. EBV-infected B cells enter the MS brain, residing for long periods of time, where they produce oligoclonal IgG bands and pathogenic autoantibodies which attack and damage neurons. Autoreactive T cells entering the brain are reactivated by EBV-infected B cells presenting CNS antigens, CP, bound to MHC. Autoreactive T cells produce cytokines and recruit other inflammatory cells that damage oligodendrocytes, myelin, and neurons. Conditions might be misdiagnosed as multiple sclerosis, neuromyelitis optica, a rare disorder that causes demyelination in the optic nerves; idiopathic transverse myelitis, spinal cord inflammation that may have a post-infectious etiology; acute disseminated encephalomyelitis, often post-infectious, causing diffuse areas of central nervous system, CNS, demyelination, fever, and encephalopathy; nutritional, vitamin B12 deficiency, zinc toxicity, copper deficiency, hypocalcemia; endocrinal, diabetic peripheral neuropathy, or hypothyroidism; vascular disease, stroke or vasculitis; AIDS, systemic lupus, erythematosus, Behçet's syndrome, sarcoidosis; infections, Lyme disease, syphilis, HIV, tropical spastic paraparesis; neoplasia, primary or metastatic neoplastic brain lesions or paraneoplastic syndromes;